I mean, that's all it's really about. It's just about doing a presentation, standing up in front of people, and like building some of those questions and realizing that it's really not that bad. It really wasn't that bad, was it? Wasn't nearly as bad as it was 2 days before. No, because 2 days before we're all freaking out about the presentation, right? And then when you actually finally give it, you're like, you know what? It really wasn't all that bad. That's all I really wanted to do was show people that it wasn't that bad. And we actually heard some pretty cool stuff. Like I like the green roofs and the.
Yeah, that was cool. That was one of my favorites. And she
She
was the one that was.
She was the one that was freaking out the most. And so she ended up doing just fine. Like there's so much more that could be talked about for green roofs, you know, like.
Yeah. I think she probably had the best presentation overall between delivery and just content. Like she did. I thought.
Yeah.
I liked it too, and I knew it would be. Like everyone did well, really, when it comes down to it. Like it's not easy giving a presentation. It's just not. And so think about how hard it is to stand up here and give an hour-long presentation, you know, and try and keep people's interest in, you know, topics and things like that. So, and they told me that shit, man, everyone's just happy to, like, we get it that you're up there struggling. We just, the more you do it, the better you get at it. And so, all it was just, you know what, like, man, that really wasn't that bad at all.
Yeah.
I saw some things where I could have improved on. I'll go make those adjustments, and then the next time it'll be better, and just grow from there. So that's all that was about. I like the vertical farming, the thought processes that were going on there, like that. Like each one of those topics could be brought as a section about
the ash spill one. That one was a good one.
The what?
The ash spill.
The coal ash spill, right? That thing could have been brought out so much more. Like that was actually part of my dissertation. Like I specifically remember citing some of that stuff. And like, man, selenium contamination anywhere around those energy plants, like in North Carolina, Duke Energy is just having they're just getting beat up left and right because they keep having the selenium contamination. Man, it was costing like $250 billion worth of damages. Right? $250 billion worth of damages. Like, it's insane what it costs to clean this stuff up. Where do we get that money from? Right? Like, it has to come from somewhere. We have to pay these people that are out there trying to, clean this contamination stuff up. So, just those are the things that we don't ever, that are off our radar. We're just trying to get to class. We're just trying to get to the exam, make it to spring break, make it to finals, and go on for summer. But man, there are some cool things out there like that. What else was cool? What else did y'all think was good?
I like Miss Kitty's. That was the subsidence.
The subsidence of the Everglades, again, a 1 hour topic.
Yeah.
That she had to condense down into 5 to 6 minutes. The irrigation one could have all been put together as like one, and that's what I kind of was hoping for, was that everyone would get together and see how to do that. Like, I think that Josh and Bren got together and they just talked about, I'm going to talk about this and you're going to talk about that. You should go first, I should go second. But like, there could have been so much more collaboration with that in there as well.
Yeah.
But man, overall, everyone did great, man. Like, just wanted people to not freak out about the presentation.
Yeah. How many do we have left?
5 or 6. Can't be that many. I haven't looked at my sheet.
Yeah, I was gonna say I've got it, but I'll have to dig it out. So.
Maybe it was 8. I'm not sure.
I don't know. I know there was
5.
5?
Okay. Yeah. I know there was 2 like before me.
Because Michaela was the last one to go last week.
Yeah, she was.
Yeah.
So who's left?
Gunter, Russell, Williams, Larson, Jones, and then they weren't here.
Curtis and.
Yep, she's got to go too.
Hewitt Corky?
Logan, he was on the baseball team.
Okay.
So 6 presentations, and then we out.
Yeah.
Michaela was like, yo. All right, so picking back up from where we left off last week, which seems forever ago, and these weeks just go forever. I think we left off with talking about the fossil fuel combustion, and that we're having this problem where producers are now having to put out more sulfur because we've scrubbed all that sulfur out of those fossil fuel combustion emissions. And so deposition is not necessarily a big problem anymore like it was probably back in the 90s. And so much like nitrogen, there is a fate for each one of these transformations. So deposition obviously can be taken up by the plant because it is a plant essential nutrient. A small portion of it can volatilize, but not as much as what we would expect from nitrogen. Again, with each one of these cycles, we're going to have some immobilization and mineralization, and it's probably going to be more adsorbed to the AEC, and then it can leach because it is a anion. And so most of our soils are negative, therefore, much like nitrate, it would leach. All right, so going back to the sulfur cycle, I don't know if y'all have this. I'll make sure that if you don't, that I post this online. But so we started out with the nitrogen cycle because it's the most complex, it has the most going on. So once I put the nitrogen cycle up and the sulfur cycle next to each other in fertility, they were like, yeah, I'm glad you started with the nitrogen cycle. So we'll have deposition, we'll have some sulfur fertilizers that can be added to our soils. We'll have some sulfur added from our plant and animal residues. Find the count. That will provide some sort of energy for our microbial populations. They need that sulfur because remember, it is part of proteins and DNA and things of that nature. So they will need that in order to survive. We have plant uptake. I think I'm just going to go from kind of top to bottom. It's just the best way that this sets up. And then we'll have a little bit of volatilization, but very little volatilization. Again, we will have our immobilization and mineralization. Sulfur has 4 oxidation states, so -2, 0, and then we can have this is going to be +6, and then SE and SO3 2 negative is going to be +4. Our sulfate can be adsorbed to the AEC, and so as our pH goes down, our AEC goes up. Correct. So we're going to find this in greater adsorption in more acidic soils, and then we're also adding sulfur, which is creating more acidity. So we'll start to have more adsorption there. And then finally, it can be leached out through the system into our groundwater. So the sulfur cycle compared to the nitrogen cycle, much more simple, but similar processes. And there are going to be similar processes in all of these. It's just the nitrogen cycle has more possible transformations. We're going to have some adsorption to the soil colloids, immobilization and mineralization. We'll have some, I mentioned adsorption to soil colloids, and then leaching because it's an anion, and most of our soils are going to be negative. So won't be held to the soil colloids as tightly. And so just a comparison between the nitrogen and the sulfur cycle. And so again, like I said, when I showed this in fertility, they were like, yeah, much easier, but still a lot of the same processes. Everyone good here? All right, we're gonna run through potassium pretty quickly. It's important, well, because the plants need it for, that's the next slide. So we have NP205, and K2O on a bag of fertilizer. So those are going to be 3 of our primary nutrients that we're trying to get to our plants. But what's most important about potassium is that we don't have to worry too much about any toxicity or eutrophication. It's just not used by those microbial populations. It's important in cellular enzymes, metabolism, nitrate reduction, photosynthesis, but most importantly, water relations. And so if we are having some issues with plants either retaining or losing water too fast, one of the first things you should be looking for is our potassium levels. And so it's kind of like when I think about potassium, I'm thinking about electrolytes and kind of like Gatorade or Powerade. Like those are the things that maintain that balance of that water potential. And then also with our leaf stomata. So the potassium is responsible for the guard cells opening up the stomata and releasing releasing that water from the plant. Good, Isaac?
Yeah.
Okay. Important in starch formation and translocation of those sugars, and this is what's going to improve the quality of our flowers, fruits, and vegetables, because we're taking up that water. It's what's giving it the color and the strengthening of the stems. And so, also when I think about potassium, I'm thinking about like the immune system of the plant and like the overall health. So if your plant has a strong immune system, it can fight off pathogens and diseases. And if it's functioning properly in like a healthy system, it's taking up those nutrients and utilizing them properly, rather than just taking them up and kind of like if you're just eating a lot of carbs but not using the energy to burn that off, right? So if you're storing more excess nutrients, there might be some toxicities. But if we have potassium that's performing the functions that it's supposed to be performing, it's keeping everything moving, that metabolism is continuously moving, and everything is going where it needs to go. We're getting the proper amount of sugars and starches going to our fruits and vegetables. This graph is just kind of showing that the concentration along the x-axis, that as the K concentration increases, that we are increasing carbon dioxide adsorption. So as potassium increases, photosynthesis will be increasing, because again, now we have this potassium that's opening up the guard cells that is allowing carbon dioxide to come in to perform photosynthesis. We get an increase in that RUBP carboxylase. If y'all had plant physiology, no? Y'all's eyes got this big, like the RUBP what? Maybe a whole different class. But anyhow, these enzymes are what is bringing in carbon dioxide and then partitioning it or compartmentalizing it to certain places to perform the Krebs cycle, things of that nature. So that whole photosynthetic cycle, the Calvin-Benson, that is going to give us our ADP and ATP. I just saw some glazing with the eyes. It's okay. Just that as we increase our potassium, we're increasing the carbon dioxide assimilation, which is basically churning our photosynthetic reactions. Again, I mentioned before that it's going to increase the resistance to fungal disease and pest tolerances and those environmental stresses. So again, like I said, this is kind of like the immune system. This is like our vitamin C and vitamin D that is, like, I guess for humans, that is improving our immune system. So potassium for plants is improving their immune system. That drought tolerance, if there's not enough water, the potassium is what tells the guard cells don't open and close, because we don't need to lose that water. And then also winter hardiness. And so if you're going to plant cover crops, it might be a good idea to put some potassium out before the cool season happens, so that you can maintain this balance and give it a chance to survive. It's fairly mobile within the plant. You'll start to see that on the edges. They'll be burned on the edges of the oldest leaves. There are 4 available forms. Well, it looks like 3 available forms. But the K that is held inside those primary mineral structures, right? And so if it's in that structure as part of this big long chemical equation, at some point, some weathering process has to happen in order for that to be released into the soil. We can have some non-exchangeable K in those secondary minerals, and those secondary minerals are easier to break down rather than our primary minerals. We have exchangeable K on the soil colloids, and then also K that is in the water solution. We can have some potassium fixation. Remember that those clay layers open up. Potassium, as water separates those clay layers, potassium fills those spaces in between, and then when the soil dries out, it collapses back over them. Some of the factors are going to be the clay mineral type and the CEC, exchangeable K, and then there are some environmental components that are going to influence how well that K is adsorbed in those inner layers. We're not going to worry about the potassium cycle because this is the simplest one of all. It's in the soil. We can add it through fertilizers. The crops are going to remove some of that. And what I don't see is immobilization and mineralization in this. I don't see any immobilization or mineralization. So our microbes are not using potassium for any of their biological functions. Just because I pulled this from fertility. Some of y'all might have remembered that potassium makes up, elemental K makes up 83% of this K2O molecule. It's not very likely that you're going to see that on a soil test result, that you have to add elemental K, because they'll end up making that conversion for you, and they'll tell you to put it out as K2O. But again, this was something that my fertility class greatly appreciated, because Dr. Earhart did not explain where 83% came from. It's a constant, but it's constant because the molecule is constant. All right, that ends it for the cycles, at least for nitrogen, phosphorus, and potassium and sulfur. And I'm going to kind of segue this into the carbon cycle and soil organic matter, and hopefully try and bring in some soil health components to kind of the reason why we're looking at this carbon sequestration, and hopefully that'll get us through most of April. And then also some of those soil chemistry lab experiments. I'm trying to find some of those, and they seem to be very basic, like soils type stuff. I don't know. I tend to like to be a little extravagant, so I want to do something cool. I want to see something separate out. I want to see something turn color. I want to see something that like is visual. All right, so for your notes, the carbon cycle is going to be those crop residues, and this is what is going to provide an energy source for our microorganisms. And our microorganisms are important because they're the driving force of our biogeochemical cycling. And so the difference between mineralization and immobilization, I don't think I've brought this up for a moment, but that immobilization is the conversion, y'all are probably going to want to write this down. Immobilization is the conversion of organic or inorganic to organic. And maybe that was in like the very first module. And then that mineralization is the conversion from organic to inorganic, which is what our plants are going to take up. They take up the inorganic form and then convert it right back to an organic form, right? So there's an immobilization by the microbes or a mineralization so that the plants can then take it up and immobilize it or mineralize it, right? So we have this big cycle that is continuing to go on. For carbon mineralization, those microbes are converting that organic carbon, they are oxidizing it, or it is becoming oxidized to carbon dioxide. And that carbon dioxide is released into the atmosphere, and we are well aware that there are some funky things going on with our weather. and we are correlating that to our changes in carbon dioxide concentration and some of our other greenhouse gases. There are two basic processes from the carbon cycle: photosynthesis and decomposition. And so we are removing carbon dioxide from the atmosphere. Plants are taking it up in that RUBB carboxylase, depending on if it's C3 or C4, is compartmentalizing that, using it for the photosynthetic reaction to create glucose that is eventually providing energy system for this plant to grow, and then during that process that the plants are giving back off oxygen, right? That's real quick, fast and dry, the way that plants work. And then finally decomposition. And so after plants die, that returns back to the ground. And so the microorganisms are utilizing the energy that is in that plant material in order to continue this carbon cycle. Not that we're really going to need to be able to draw the carbon cycle, but we do know that there's going to be some volcanic eruption that is going to have some carbon dioxide in it, methane. Remember from the video that we watched with the great oxygenation event, that we had a lot of methane in the atmosphere. Eventually we added oxygen, and that methane was then oxidized to carbon dioxide. We'll have some photosynthesis and some respiration. Every once in a while we'll have some more carbon added back through combustion. And if you remember going all the way back to the first law of thermodynamics, that energy cannot be created nor destroyed. The same goes for matter. Matter cannot be created or destroyed. It has to go somewhere. It doesn't just disappear. We have some peats and fossil fuels. This is going to be our coal and our peat bogs. And then we will have our decomposition of whether it be plants or animals, and even human remains as well. So those are all contributing to this cycle that is continuously moving, but it is the microorganisms that are driving that. And so the majority of the work these days are really focused on carbon sequestration. And I think that that is going to be something that will help y'all as you move out. So I think, again, this is setting up kind of April, where we're going to have a little bit more talk about this carbon sequestration and soil quality and soil health, how we go about improving our microbial populations, and some of the things that can be done in order to achieve this carbon sequestration. But then our largest carbon sink is going to be the soil. We have forests, roots, and microorganisms. Y'all too might remember this from soils. They're recalcitrant, meaning stable or kind of difficult. We call, like, coal is hard to, it doesn't break down easy. It requires a lot of energy. It has a lot of energy stored, and that's why we use it for fossil fuel combustion, because of the energy that's in it. It's very stable and resistant to decomposition. The organic components of our soils are going to be those fresh residues. And so this might be a fresh residue, could be at the end of the growing season. We run a bush hog across our cornfield and chop up that carbon, and then that residue is left on the soil surface. It could be that during the growing season there's a plant that senesces, a leaf senesces or falls off, or something that is newly added to the soil organic components. There's organic matter that is decomposing. And this is where we're having issues with our tillage, and why we're not wanting to do that, because when we till, we are introducing oxygen. That oxygen is interacting with that carbon, the organic matter, and then is giving off as carbon dioxide. So if we keep it no-till, we're not introducing that oxygen, and there's more carbon that will be coming in, and we're starting to increase that organic matter. We have stable organic matter, which is going to be our coal, things of that nature, and then finally our microbial populations. And then right here, that our soil carbon is greater than both the plant and the atmospheric carbon combined. There are two fractions for soil organic matter. We have the active fraction and then the passive fraction. The active fraction, which as the name might kind of give the clue away, that this is what the microbes are actively decomposing, or they're using as the energy source. This is going to be those fresh residues that are being added to the soil. About 10 or 30% of that is responsible for, like, that's the food source. That's what the microbes are actively trying to find. Some of y'all, so y'all might remember from soils, and I know that Michaela might remember from fertility. This is kind of where those two classes would have combined, because I did overlap a lot of that. And so for carbon to nitrogen ratio, 15, 20, 30, the carbon nitrogen ratio around there, we're going to start to get either the carbon is going to be immobilized, but when we get below 50, a lot of books are going to have a lot of different numbers for it. I can never find one that agrees. Some call it 20. When the carbon nitrogen ratio is greater than 20 to 1, you'll have immobilization. I have a slide up here. The next one is that it's 24 to 1. Greater than 30 to 1, you're going to start having net immobilization. If you get less than 15 to 1, it's mineralization. And I just really think that's going to depend on A, the microbial populations, and then B, like the type of plant material. And then the passive or the slow fractions, and these are going to account for about the other 60 to 90% of that soil organic matter. And this is also going to be where our pH-dependent charge is. This is those functional groups of organic matter, and that's why our CEC and our water holding capacity increases when we have an increase in soil organic matter. So our decomposition processes, plants are composed about 75% water, 25% dry matter. On the next slide is where we'll be talking about the different components. I'm pretty sure that if you've had anything from Dr. Fenewald or any type of ruminant, this cellulose, lignin, and hemicellulose are going to be important in digestibility. As he talked about that in forages,
I think so.
slightly kind of. That's something new that I am coming up with as far as like acid detergent fiber and neutral detergent fiber. Way over what I got going on. I grow corn and then we're going to harvest it. So anyhow, what's important to note about this slide is the chemical makeup of these compounds. And so here you can see we have we're starting to have, anybody had organic? Don't do it. Keep your GPA. We have these bicyclic phenolic compounds, and you start to see that that as these compounds get larger in molecular weight, they are more difficult to decompose. It takes a lot of energy to break those phenolic rings. Not so much to break these bicyclic rings, but for this lignin, it's very difficult to break that down. Yeah, that's something you've encountered before?
We've talked about that in chemistry before.
In chemistry, right. And so this is kind of real world practical application as to why that it takes a lot to break down that phenolic ring. That's why this is the slowest to decompose. So I'll talk about this on this slide. So while we want to say that there are differences between plants and humans, it's very easy for me to relate back to the thought processes that go along with plants as to what go along with humans. I know y'all seen the commercial, you're not yourself when you're hungry, you have Snickers. That's because those sugars and starches and the proteins that are in that Snickers bar are easy to break down. And so you can, have y'all ever gotten shaky and been like, I need to eat something now? Like I can feel my blood sugar off a little bit, so you grab some Skittles or a bag of peanuts or a Kind bar or something like that. That contains sugars and starches and simple proteins that your body can utilize very quickly. Those biochemical reactions happen quick enough such that it stabilizes your blood sugar. Next are going to be crude proteins, and then we have the hemicellulose and cellulose. I'm going back from, I'll see y'all right. Those are going to be those bicyclic compounds that are difficult to break down. Fats and waxes are difficult to break down. It's just because they require a lot of energy to break those long chain fatty acids, right? Y'all have heard those, the long carbon chains. Again, going back, those carbon bonds are difficult to break. And then finally our lignin and our phenolic compounds. So here we have I want to make sure I got this right. All right, so we're looking at hemicellulose, cellulose, and lignin. And so that hemicellulose breaks down quicker than cellulose, and then cellulose takes longer than lignin. Where are we going? Lignin being our largest weight molecule, hemicellulose and cellulose being lower weights. It's all based on the molecular weight of that molecule. Simple sugars and proteins like C6H12O6, glucose, is broken down fairly quickly because it has to in order for the plant, in order to be able to use that. That's also why we need more of it. That's why we're conducting photosynthesis and all of those processes so that the plants can grow. All good here? This is probably y'all remember this question from soils? This was a rank them in order of rates of decomposition. But again, if you can.
Multiple questions of that?
What's that?
I think there were multiple questions of decomposition.
Yeah, I think I just kind of had them spaced out, as in like one was, I think I remember one of them being list these from fastest to slowest, and then I flipped it around and asked them from slowest to fastest. Which one would decompose quicker, hemicellulose or cellulose, things of that nature, just to understand that our proteins are going to be quickest, cellulose in the middle, and then our fats and lignins and phenolic compounds. So we have two types of decomposition, aerobic and anaerobic, with and without oxygen, right? And this decomposition is essential for the biogeochemical cycling of our nitrogen, our phosphorus, and our sulfur. Some of those compounds that are formed can be very resistant to the microbial action. So there are people who study this type of stuff, and they know exactly how fast a certain material will break down and how long it takes to decompose. Again, they took a lot of, I'll bet you they took a lot of biochemistry and a lot of organic chemistry. Not what we do here. Not that you need to know this formula, but that there's going to be some energy released associated with this, right? So that we're going to have this oxidation because we are in an aerobic environment, and that there's going to be some energy given off from this. And so going all the way back to almost like one of the very first or second lectures that we had, I had that equation that had Gibbs free energy equation. This is going all the way back to it. And so what they'll do is they know how much soil chemists can start to calculate how much energy needs to be inputted into that system in order to break down those lignin molecules. They know the energy of each one of those phenolic rings. They know how much energy it takes in order to change those inner sphere and outer sphere complexes. How do we adsorb and desorb these cations and anions onto or off of a soil colloid? That's really all that is. Not that y'all will ever be calculating any of that. That's why we really didn't go over that. Just to understand that this was a Gibbs free energy equation. And they know that we're going to have this amount of energy that is given off. That's what the microbes are receiving. So we talked about the lignin being the hardest, the most difficult to decompose, but we do have friends in low places. And these are going to be our white rock fungi, and they're the ones that have enough energy or that don't require large quantities of energy in order to live throughout the day. I guess you could kind of figure it like that. Like we're supposed to eat 2,000 calories a day in order to be functional. And if we're thinking about that, like they may only need 100. So they don't require large energy requirements, and so they can sit there and chew on this lignin for a while, and they also know that they have it, that this substance is going to be there for a minute, and they're not worried about it because nobody else can break it down either. So there's no competition among these microbes for this lignin molecule. And then anaerobic decomposition is going to be much slower because there's no oxygen, and those microbes do need that. They do need oxygen in order to perform this respiration. And if there's no oxygen, well, then there's not a whole lot of activity. And so most of those byproducts are going to be methane, where we go from carbon dioxide going from carbon dioxide to methane is oxidation or reduction? A reduction. Because the microbes are using this oxygen, or they're using the electrons that are associated with this molecule. So the byproduct that they give off is methane. And we know that methane also gives off those greenhouse gases. Just like from our denitrification process, those greenhouse gases are going to be what is causing this global climate shift, change, warming, however people care to perceive that. two major organisms, or two classes, I guess, of organisms. One is going to be autochthonous, and they are going to digest that very stable, recalcitrant soil organic matter reserves. Maybe not that they're sitting there chewing on a big chunk of coal, but they are decomposing that lignin, the more difficult carbon source. Next we have this zymogenous class, and they're the microbes that start to attack leaf litter, or senesced leaves, or twigs. Yeah, maybe not the twigs, that might be a different species, or a different class, because twigs have a lot of lignin in them because they're more tree than they are plant. That's not correct. These are going to be our high activity bacteria and actinomycetes, and they'll be decomposing the cellulose material. So I'll wait until y'all get finished. So, zymogenous bacteria that are going to be able to break down those sugars and starches very quickly, and then our autochthonous down at the bottom, those very hard to decompose materials. Those rates of decomposition and mineralization are going to be associated with environmental conditions. If it's cold outside, the microbes are not playing. They're kind of in hibernation because it's not warm enough. Those metabolic functions don't happen as quickly. But then as we increase our temperatures, and going all the way back from soils and maybe even chemistry, that for every 10 degrees Celsius increase in temperature, we get a 2x increase in biological rate. I saw you trying to go back through the Rolodex, right? But just that as temperature increases, the biochemical rate increases, like in a nutshell. So that we're going to have more of these decomposition processes happen in the summertime rather than in the winter. That's why when the leaves that fall in the fall, they don't decompose until the summer comes. We get temperature, we get rainfall, and in our gutters, there's not very many microbes up there. So that decomposition is a lot slower when we get that nasty, mucky, half decomposed, half not kind of. So environmental conditions, the quality of the residues. So what is the makeup? There are going to be some physical factors that are going to come into play, and then those carbon to nitrogen ratios. Quality of residues. This is why this slide was so important earlier, right? If we have our sugars and starches, they're going to break down a lot quicker. If it's a tree, it takes a long time for a tree to decompose, doesn't it? So all this slide is really kind of mentioning. Physical factors are going to be primarily our location and like our particle size. If a leaf falls on top of the soil, then the microbes that are under the soil don't have an opportunity to decompose it. Pretty simple. And so then some of those nutrients that are inside that leaf material can then be introduced into our waterways. And I was mentioning this, I forget which class I was mentioning it in. You ever see the the lawn care people, and they come and they cut your grass, or they come cut a grass lawn, and it's in the streets, and they all blow it towards the gutter. And then they blow it into the water system, and that water system now has to transport those leaves back to the waste treatment facility. That's what I think of when I think about these nutrients are susceptible to runoff. Like, if we don't keep those on site where the microbes can break those down, they go into our waterways. And then the smaller size, the greater the surface area. Also, that if we cut a leaf, that allows that microbe or that bacteria to actually get inside the leaf and start to break it down from the inside. This was where I was talking about that no book that I've read has ever been, they just can't agree on it. But I think that most of the scientists would agree that somewhere between 24 and 25 to 1 is the ideal microbial diet. That's where everything is in enough balance that we are not getting a net immobilization or net mineralization. But that as we start to move down below 20 to 1, in this range, we will have a net mineralization because there's more nitrogen than there is carbon. And that nitrogen is converted or mineralized from organic nitrogen to inorganic. But then as we get greater than 25 to 1 and we start getting into like 30 to 1 and higher, the microbes will then take the nitrogen that is in the soil as ammonium or nitrates or inorganic nitrogen and start to convert that to organic nitrogen in order to break down these substances, in order to break down these materials. And so that as our carbon content increases, our lignin content is likely to increase as well. Because remember, it's those phenolic rings, and each one of those lines are carbon, so the molecular weight starts to increase as we increase our carbon material. Anybody's grandparents compost? Grow a garden? So my grandfather would rake up all of his leaves out of the yard and then put them in the garden. And then he would go to the stables and get horse manure to put on top of the leaves so that there was enough nitrogen that the microbes could break those leaves down. Because if not, the leaves would just sit there. It's just like watching a sawdust pile. It's not going anywhere, right? And so finally, over time, these plant materials turn into humus. Not like the hummus that we eat on crackers and sour or whatever that is. This is going to be our fulvic and humic acids and humin. It's important to note that this humus decomposes at a rate of about 2.5% a year. And so we want to make sure that we're at least putting 2.5% back or more. But again, it's going to depend on the types of crops that we're putting back into the soil, because those microbes can't break down lignin as fast as they can starches or cover crops or some of those softer, more nitrogen